Hello, I'm Kimberly Glass, and I'm really excited today to talk to you about the principles of network science, which I think is one of the most fundamental topics, kind of laying the groundwork for the course today. Before I get going, I'm going to warn you there's going to be a lot of terminology in this lecture, but I think it's all going to be really important. And hopefully, by the end of the course, you'll understand why it was important to learn all these words and terms. But before launching into that, I want to start with the idea that networks are all around us. And this is really, really critical because networks aren't just encountered in biology, they're encountered in our everyday life. This right here is a picture of the internet circa 2021, where you can see color coded based on connections in the internet, the wiring, the physical lines that connect different routers in the internet in different areas of the world, and the backbone, the spine of the internet that connects the entire globe together. And so today, as you watch this lecture, you're actually taking advantage of a network. You're taking advantage of the fact that I can connect to you virtually from somewhere else via video. This is another network, a social network, that is commonly analyzed in the context of network science because it really well illustrates the idea of structure in network. And this particular network, they queried middle school students about their friends. And every connection between these circles in this represents when one student said they were friends with another student. You'll notice right away that this network has a peculiar structure to it. It has a left and a right, a top and a bottom. It turns out that the dots on the left are the students who are Caucasian. The dots on the right are primarily the students who are African American. Those are the green dots. And the red dots, those are Asian students. What about the top and the bottom? The top, I believe, are seventh grade students, and the bottom are eighth grade students, showing that there was a lot of connections between students in the same grade, but also between the same race. Tells you something very striking about our social patterns, or at least the social patterns in this particular middle school. This is another small illustration of a network that's commonly illustrated in the context of network science. This is a sexual contact network. And what you can see with this network is there's some people in this network. These circles represent people who are hubs, who have many, many different partners. And some people who are have only one partner, right? Or only a handful of partners. And why this type of network can be really, really important is if you want to do something like mitigate the spread of something like AIDS, a disease that spread through sexual contact, you may want to target someone who's a hub in the network or someone who really breaks off a portion of this network, right? That connects disparate parts to prevent the disease from spreading. Many of us may have even traveled on a network today to wherever we're at. We may live along a network. On the left here, this is a map of the T transit system in Boston. On the right is the metro system in Rome. And what you can see is this itself is a network. Each individual stop along the tracks can be thought of as a node, with connections being thought of as edges. You have certain stations that, for example, that are downtown that are much more important in this particular network structure. So for example, if the Termini train central station in Rome goes down, the entire network may fall apart. Similarly, the Park Street or downtown crossing in Boston, if something happens at those particular stations, the entire network falls apart. However, things that are on the terminal, those may be less important to the overall structure. Similarly, I'm going to show you another very important network. This is another transportation network, but it's a transportation network of flights, of connections between different airports. Now, why is this particular network important? This network is important is because it's highly studied in the context of epidemic spread. Having recently been through the COVID-19 pandemic, we can understand the importance of this type of network, right? Where people would shut down flights, they would limit connections across the globe, attempting to prevent the spread of COVID-19. So these type of transportation networks can be used to study epidemics because of this notion of things flowing along the network. Now, there are many types of networks that are encountered in molecular biology, and I'll touch on some of those at the end of my talk. Very briefly, you might run into things like protein-protein interaction networks, protein-D interaction networks, genetic interaction networks, metabolic reactions, co-expression interactions, text mining interactions or association networks, and many, many more. There's no way I could cover all the different types of networks encountered in molecular biology today. But before we even begin discussing these types of networks, we need to know, what is a network? We need to define it. Now, in its very simplest format, a network is a set of nodes connected by a set of edges. Over here on the right, you can see a network composed of 1, 2, 3, 4, 5 nodes. Those are these circles. And 1, 2, 3, 4, 5 edges. Those are these lines. A subnetwork is a subset of the full network. It is a subset of the nodes in this full network and all the edges between them. So here you can see node 1, 2, and 3, 1, 2, and 3, and the edges between them form this subnetwork, which is a portion of this full network. Now, importantly, you won't encounter that probably in this course, but if you get really interested in network science and start reading on your own, you'll note that in some fields, a network isn't called a network, but instead it's called a graph. A node isn't called a node, instead it's called a vertex. In fact, that is the history behind this graphic here. I borrowed this graphic from someone with a history in computer science, and he didn't call this node 1, he called this vertex 1. Hence the history and the important clarification. So what are some common network structures and features that you may encounter? Well, first, most commonly, you may have a network that is undirected, as I illustrated before, that's just a single line connecting two nodes. But a network may also be directed in that there may be information that only flows one direction along the line from this top node to this bottom node. Networks can also be signed. For example, in this case, you may have a repressive interaction where information is flowing from this node to this node, but in a repressive or negative way, or for example, a positive way. So a network can be signed, or in this case up here, unsigned. Networks can also be weighted. In this case, each edge is given a value or a quantity to illustrate something about the connection of the edge of the association between the two nodes, right? So this edge has a weight of 6 and is much stronger than, for example, this edge right here that has a weight of 0.5. And importantly, these weighted edge networks, they're generally positive, but of course, weights can also take negative values, which means they can be both signed and weighted. There can also be cycles in networks, and these are important network features. In the context of an undirected graph, you just look for simple loops. In a directed network, this will go in a cycle, a circle like this. This leads us to the idea of an acyclic graph. And in the context of a directed network, this is often referred to as a DAG or a directed acyclic graph. Some other less common but still important network features to be aware of. The idea of self-connections when a node connects to itself is commonly called a loop. A multigraph includes edges, multiple types of edges between nodes, illustrated here as a blue edge and a red edge connecting the same set of nodes. That's a multigraph. You can also have things like bipartite networks. In a bipartite network, there are nodes of two types. In this case, you have blue nodes and orange nodes, with edges extending between nodes of different types, but not between nodes of the same type. One common network feature is this idea of network density, and it's actually really important when thinking about how to analyze a network, how dense it is. So network density is simply defined as the number of edges in the network divided by the number of possible edges. And an undirected network, the number of possible edges is the number of nodes in the network, choose 2. And a directed network, the number of possible edges is simply the number of nodes squared. A network is considered sparse if the number of edges is approximately equal to the number of nodes. So for example, up here on the upper right, you have 1, 2, 3, 4, 5 nodes, and 1, 2, 3, 4, 5 edges. This is a sparse network. A network is considered dense if the number of edges starts to approximate the total number of possible edges. In the case of the undirected network, Of course, this is number of nodes choose 2, and the directed network is the number of nodes squared. This is an example of a dense network. It's not all the edges, but many of the edges are there. A network is considered complete when it is completely filled in. Every single edge that could exist does exist, right? So for example, in the underdirected edges network, the number of edges is equal to number of nodes choose 2. So the number of edges is equal to the number of possible edges. Same basic idea for the directed network. Now, importantly, as an idea of a terminology, a clique is a fully connected or a complete subnetwork, right? So this could be considered a clique if it is a complete subnetwork where there are other edges maybe connecting out to other portions of the graph or network. So how do we describe network connectivity? The most basic way to describe network connectivity is through the idea of connected components. Here I show a toy network with 69 nodes and 71 edges. This network is composed of 6 connected components. 5 of those are circled here in green. 1, 2, 3, 4, 5. And then you have this largest one right here. This leaves us the idea of the largest connected component. The largest connected component is the connected component in the graph that has the most number of nodes. If the graph is highly fragmented, the largest connecting component can be fairly small. But for most commonly encountered networks, the largest connected component actually takes into account a significant fraction of the nodes. And it can, in this case, also be called a giant component. How else can we describe network connectivity? The idea of network paths. A path is a sequence of edges in the network. For example, this is a path from this node to this node to this node to this node. A closed path on the network is called a graph cycle or a circuit. Why is this important? Well, network paths can tell you something about how you can traverse the network to get from one point to another. And that leads us to the idea of shortest paths. The shortest path is how you can connect 2 nodes by as few of steps or edges as possible. So for example, the shortest path from node A to node B is 4. 1, 2, 3, 4. The shortest path between node C to node D is 10. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. The network diameter is the longest, shortest path in the network. It tells you something about the size of the network and the sense of how many steps you need to take in order to get between any pair of nodes in the network. And this particular network, the diameter is 14. The longest, shortest path in this network goes from 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Finally, this leaves us the idea of betweenness, which is a measure of node centrality, how central a node is to the network. The betweenness is defined as the number of shortest paths through a node or edge. It can be very challenging to calculate just by eye, and commonly done computationally, but here is an example node where we can easily see the betweenness is equal to 3. So in this node, you have exactly 1, 2, 3 shortest paths going through this node that allows these other nodes to all connect to each other. Another common measure of node centrality, or measuring node connectivity, is the degree. The degree is the number of edges associated with an individual node. So for example, this node here has a degree of 1. It has exactly 1 edge connected to that node. This node has a degree of 6. It has 6 edges connected to the node. Importantly, the idea of degree is different in an undirected versus a directed network. In the underdirected network, this node has a degree of 6, but in the directed network, this node has both an in degree and an out degree. In this case, the in degree is 3, 1, 2, 3 edges pointing in, and the out degree is 3, 1, 2, 3 edges pointing out. If people ever talk about the hubs of a network, you should know that a hub is simply a node with a large number of edges. There's not a clear formal definition, but it's basically one that has more edges than any other node in the network. The network degree distribution is another important property of a network because it tells us something about the overall structure of the network. So for example, in a random network, you can look at the probability of an individual node on the y-axis has a particular degree on the x-axis. In a random network, this is basically Poissonian or Gaussian, as a nice little bell-shaped curve right there. However, in many real networks, including the ones encountered in network biology and medicine, you often find that the degree distribution follows a power law. The probability of a given node on a log scale is a line when you look at the degree also on a log scale. And it's right here, this is the equation. Such networks are said to be scale-free. You can see the difference in structure between the random and the scale-free network here. Now, another important concept related to the degree is this idea of network mixing or sortativity. And this is a very cool feature in networks. In an assortative network, nodes tend to connect to other nodes of similar degree. Things like authorship networks can sometimes follow this type of pattern. People who have a high number of co-authors also tend to connect with another number of high co-authorships, right? You can see this in this table over here. A disassortative network, nodes tend to connect to other nodes of dissimilar degree. So for example, a high degree node will connect to many low degree nodes. Very importantly, based on this table from a PRE paper in 2003, a canonical paper in which they studied many biological networks. Many biological networks are known to be disassortative, meaning that hubs, very high degree nodes, tend to connect to many low degree nodes, say nodes with just a single connection, the one to the hub. This leads us to the importance of things like bottlenecks and information flow, as illustrated from this figure in this paper here. And this particular paper, you can see that there are different types of nodes. For example, you can have a hub node that is also considered a bottleneck, meaning that information must flow through this green node, right? It's a bottleneck because information must go through it. It's also a hub because it has many connections. You can also have non-hub bottleneck nodes that are important for information flow, but don't necessarily have a large number of connections, such as this one here. You can also have hub nodes that are not bottlenecks, such as this one here, and all other nodes, which are not hub and also not bottlenecks. So finally, let's talk about community structure, because this is highly related. Network communities are sets of nodes within which there are many more connections than expected by chance, but between which there are many fewer. So for example, in this toy network illustrated here, there are 3 communities. Within each of these communities, there's a set of nodes between which there are many, many more connections than one might expect by chance. But between these different communities, the red, the cyan, and the blue community, you only have a handful of connections. This network has a high, what's called modularity, and it has a high community structure. And in biological networks, these communities might represent things like shared function, shared groups of proteins that tend to cooperate together, etc. So in the context of biological networks, we tend to derive networks from biological data. These types of networks aren't generally known a priori. And there's many types of biological data that has been used and represented in networks. This can include things like gene expression, physical interaction data, genetic interaction data, colocalization data, sequence data, etc. And I'm going to cover a handful of the most common types of networks encountered in network medicine going forward. But before I do that, I'd like to talk a little bit about what the basic structure of biological networks is. In a biological network, the nodes can represent many different things. Typically, they represent things like genes, proteins, metabolites, biological functions, etc. Connections or edges between those nodes tend to represent things like gene-gene interactions, gene-protein interactions, etc. And importantly, these edges can represent many different types of relationships, such as gene regulatory relationships, physical protein interactions, functional relationships, etc. So what types of networks are generally counted in network medicine? One of the most common types of networks we will encounter in network medicine is the gene co-expression network. These networks are derived from gene expression data. You can imagine gene expression data as a matrix in which genes are on the rows and samples on the columns. A gene co-expression network can be derived by simply building some sort of correlation metric between every single pair of genes. Now, this by itself is not necessarily a network, so often there's some additional processing that happens. So for example, this is just a toy illustration of what a gene co-expression network might look like. In this case, each circle represents a gene. The edges between these genes are connections that represent whether or not their expression levels are correlated. Now, in the context of something like Pearson correlation, this correlation might be either strongly positive, illustrated by the red edges, or strongly negative, illustrated by the blue edges. And of course, when building a gene co-expression network, one needs to decide whether or not you're considering all levels of high correlation, both positive and negative, in which case you need to be analyzing a signed network, or if you're more interested in something like the absolute value of the correlation, or only positive or only negative correlation. Importantly, even though this is commonly used in the context of gene expression data, correlation-based networks are highly used across all types of omics data that's commonly encountered in the field of network medicine. In this case, the basic approach is the same. You have some sort of omic data in which the rows are molecules, the columns are samples. You build some sort of correlation matrix, illustrating the association between pairs of molecules, and then threshold, analyze either signed or unsigned networks, etc., based on this correlation measure. Here are two papers that I found that had really nice illustration of correlation-based networks and how they were used. They are no means the only papers that are out there. On the top left, these authors built gene co-expression networks, had edges between pairs of genes based on positive and negative correlations, and they were able to color code the genes based on their particular functions and how well they correlated with other nodes in the network. On the bottom right, this is a metabolic correlation network. In this case, the nodes are metabolites, and edges illustrate high levels of correlation between those metabolites based on data. Biomarker networks are another place where correlation-based approaches have commonly been used. So for example, on the left is a partial correlation network between biomarkers and COPD, where the biomarkers are measured across a series of subjects with COPD, and the co-occurrence of these different biomarkers was measured based on partial correlation. On the right, similar approach, except for this was based using mutual information, and the biomarkers are in the context of diabetes. And you can see that there are some times in which the different biomarkers are connected, and the different colors, dashed, etc. lines represent different contexts where there is a high level of mutual information between these biomarkers in one context compared to another. Another type of network commonly encountered in network medicine is gene regulatory networks. So what is a gene regulatory network? A gene regulatory network is derived of the notion of transcription factors, which are a type of gene that encodes a regulator. And these transcription factors regulate downstream target genes. This is a directed network, which always go between transcription factors and their downstream targets. Now, these target genes may themselves encode other transcription factors, leading to cascading processes, right? One transcription factor regulates another, regulates another, regulates another, and this really leads to the notion of biological processes. Now, not every single gene encodes a transcription factor. There are genes that encode other types of proteins that are very important for cellular processes outside of gene regulation. And this leads to a very particular structure to gene regulatory networks, where you have genes that encode transcription factors, shown here as circles, noted with a TF. These nodes have both in and out degree. And you also have nodes in these networks that only have in degree, right? They are not regulators themselves. They encode other types of genes. Here are two example papers studying gene regulatory network. On the paper on the left, the authors studied the difference between angiogenic and non-angiogenic subtypes of ovarian cancer. The colors of the edges represent whether or not these regulatory networks were found in the angiogenic subtype or uniquely to the non-angiogenic subtype, shown in red and blue respectively. On the right is a paper looking at networks in colon cancer in the context of males versus females. And the edges represent whether or not edges were found uniquely in females or, for example, uniquely in males, showing that there are different types of gene regulatory patterns in different sexes. Another common type of network encountered in network medicine is the protein interaction network. Now, proteins do not act in isolation in the cell. So, for example, when genes are regulated, they form a protein complex, as illustrated here on the left. This can be used to derive a protein-protein interaction network. So for example, the complex shown here on the left might have this structure in a protein-protein interaction network. Edges in a protein-protein interaction network exist if the proteins physically interact. So for example, here, this protein is physically touching or interacting this pink protein and the cyan protein. And over here, this mauve protein interacts with the pink and the cyan protein. This purple protein only interacts with the pink protein, one edge here. So you can have this complex structure here illustrated in a network. One classic example of use of protein interaction networks is this paper by Jorge Mensch that came out in Science in 2015. And what they took the proteins that are encoded by genes commonly known to be involved in 3 different diseases, and they put them on a known protein-protein interaction network derived from data. And they found that proteins that are associated with the same disease, illustrated here in red, yellow, and blue, tended to be much closer proximity to each other in the protein-protein interaction network than you might expect by chance. Another important type of network is the genetic interaction network. This network is derived from synthetic lethality data. In this case, you have an example where expression of 2 proteins in wild type leads to a functioning cell. This is your wild type cell. If you knock out one protein, that cell is still viable, meaning it's still alive. If you knock out the other protein, the cell is still viable, meaning it's still alive. But if you knock out both proteins, you get cell death. In this case, you have an edge between, for example, this mauve and cyan protein, and it indicates that knocking out both proteins or mutating them causes lethality. It causes cell death. And this is a corresponding paper analyzing a genetic interaction network that was published in Science in 2010. It's a landmark paper in the field. This is the Human Genetic Interaction Network. And what the authors did here is after they mapped out the genetic interaction network, they determined the functions of the proteins of different groups of functions. So for example, you have things like secretion and vesicle transport colored here in yellow. And you can see proteins of similar function tend to be all clustered together. This illustrates an important measure of robustness and redundancy in this network, meaning you can lose perhaps one of these proteins and it's fine, right? They all still have other functioning partners that do similar functions. But if you use a pair of them, then the cell starts to die because it can no longer do this particular function. Another landmark paper published in PNAS in 2007 mapped the network between diseases, right? So this is a case where a node in the network is not a biological molecule, but still illustrates something very important for medicine. So in this particular network, these circles represent different diseases, and connections represent common genes that are known to be important for that particular disease. One of the most prominent clusters in this network is right here, shown here, and I would call this a cyan, a dark blue. This represents cancers, right? So here's leukemia, colon cancer, lymphoma, breast cancer, prostate cancer, etc. And what you can see is that even though different cancers have different underlying mechanisms, they arise in different tissues, they also tend to share some of the same underlying genes. These genes regulate processes that are important for cancer development, things like uncontrolled cell growth, a loss of cell cycle checkpoints, etc. Another fun example I'd like to point out are networks of foods. Over here on the left is a cute little example where the authors went through and they determined the different nutrients in different foods. And they found that, for example, these low-calorie foods, all illustrated here, tended to share common nutrients, right? In this network, nodes are foods, edges represent common nutrients. And foods with similar nutrients tend to cluster together in this network, illustrated similar nutritional profiles. On the right, these are networks that were constructed using food frequency questionnaires given to people approximately 10 years before they may or may not have developed Alzheimer's disease. And what you can see is for people who went on to develop Alzheimer's disease, the profile of their co-consumption of food, which is what these networks represent, was distinctly different than those that didn't go on to develop Alzheimer's. In the case of people who develop Alzheimer's, you had more connection between something like potatoes and bread, crisp bread and jam. And people who were controls, you had potatoes linked with things like coffee, right, and fruit juice. And it was very fascinating because when we looked at the food frequency questionnaire, you didn't see large differences in the total amount of consumption, but you did see striking differences in the co-consumption patterns. Finally, the last thing I want to point out is this really important notion of a multi-tier multiplex network. This is a figure from this very commonly cited paper about multiple omics layer in Trends in Biotechnology from 2016. It basically makes the point that conventional molecular biology tends to follow the central dogma. You have things going from the genome, the transcriptome, the proteome, etc. And this is a central dogma where things are connected through different types of omics, but it's one single connection. Network medicine and single omics molecular network analysis tends to focus on individual omics layer. You may build connections between pairs of entities in a given omic, for example, between different locations on the DNA, between different genes based on their expression patterns, based on connections between different proteins, based on protein interactions, etc. Going forward, it's going to be really important, or at least according to the authors, to build connections not only between network layers, but across network layers, understanding the connections both in relationship to the central dogma as well as individually measured by the different omics data. All right, I would like to spend the last minute just summarizing, kind of wrapping up to kind of illustrate some of the key points I hope that you have taken home today. First off, I hope you realize that networks are everywhere. You interact with them in your everyday life. And I hope that as you walk out today, you're not only going to be thinking more about networks in biology and medicine, but also the network connecting you to every other individual in the world through your cell phone, through transportation, etc. Network approaches provide a toolkit to understand and quantify the structure found in complex relationships. There's a lot of terminology that I went through at the beginning of this talk, but it's all very important, very illustrative of how we can actually use networks to uncover different types of structures that we wouldn't be able to see without these types of approaches. Importantly, it's important to know that network can be thought of in terms of three different things: their nodes, their edges, and what those edges represent. And this is incredibly important in the context of biological networks and the networks we found in network medicine, because those type of networks can represent many types of biological data, and they are used to gain insights into many different types of problems in biology and medicine. And in order to really understand these networks, we need to know what their nodes are, their edges are, and what they represent. I hope that today I've given you a toolkit to understand all the different terminology, the types of networks you're going to see over the next couple of days of this course. I'm looking forward to our panel discussion tomorrow and your questions there. Thank you very much for your time, and I'll see you tomorrow. Thank you.